So I'm really happy to present our first keynote speaker, Dr. Soran Gerstein. He's a professor of pediatric research at the University Center Hamburg. And his research is really focused on, inborn errors of metabolism. And I just would give you a really brief introduction to allow him to speak himself, that maintaining very healthy proteostasis, proteostasis is homeostasis of proteome, is really essential for healthy state of whole our body. It's not just brain, not just heart, it's for any tissues that we can imagine. So when we talk about proteostasis, we think about different mechanisms, about protein synthesis, about protein degradation, and about protein translocation, and all these processes are tightly regulated, and during the last, I think, 20 years was enormous amount of work trying to understand how this whole network, it's really a network, it's not a single process, is regulated in different types of tissue. And for me it was really exciting to see that, for example, neuronal activity takes a very active role in this process, something that we completely did not know before. So, and in relation to our meeting today, we will see in the talk now, right, about new conceptual and like realistic way to fix malfunctioning of this network, right? And I think we will listen to the talk describing different ways and the synthesis of pharmacological chaperones, right, that really help to protect our proteins and conformations in a good way against misfolding, okay, trying to make it simple for everyone. So thank you very much for coming, and we are waiting for your talk.
Good morning. Thank you very much for the invitation. I have already learned a lot, and I very much appreciate the spirit this conference is held in. So we heard about convergence, we heard about translation, and we heard about the problems, how to bring people into the direction of drug development that come from basic research, who maybe in the first line do not think about it, to go to application of their ideas. And then obviously Dana cares for the process of how to translate good ideas and first proof of concepts into into the market later. And I think this is one of the big problems we have. Now I maybe have to switch here. Okay, thank you. If you give me a sign where the... this looks good. Good. Does it work? Very good. Today I would love to of course give you a little trip through, I mean, first the medical needs, of course, and then why we do all these projects, but also how to come, yes, worked out, how to come from the mechanisms to the drug. And I think we try to do all this trip in 12 years now, and 12 years is a long time, but 12 years ago we started knowing nothing, just starting with an idea. And I think with all the techniques coming up, we are getting faster and faster, and also the money that is needed to come to the end point will be less and less. So I will focus a little bit on why we need academic drug development. I think that everybody here in the audience would say, yes, we need academic drug development. Then I talk a little bit about the loss of function in genetic diseases. I present briefly the Kinder-UKE screening platform and the results from our 2 screening campaigns. So why do we need academic drug development? I do not know whether Israel is part of the IRDIRC consortiums. It is, okay, certainly. So there have been these goals, Horizon 2020 and IRDIRC, so what to do for rare diseases. And they wanted to find out until 2020 the genetic causes of all 8,000 genetic disorders. This is not yet accomplished. Okay, diagnostic tests for only 6,000. I do not understand this difference, but okay, maybe they thought that this is too optimistic to have 8,000, and new therapies, 200 new therapies in 2020. If we look in the literature, the cumulative number of new rare diseases by month since 2010, we see that there's a steep increase. And this is certainly due to the fact that we have whole exome sequencing and other novel techniques. The same number or the same increase goes for the genes linked to rare diseases. So we know the genetic causes. Also, the genetic tests that are available until 2017 is this representation. Now we have more than 5,000 genetic tests, so we will reach this goal. And one goal was already reached ahead of time. This was that we had 222 new therapies for rare diseases already in 2016. However, we should have a look what these new therapies are targeting. And as you said, I'm focusing on inborn errors of metabolism, so we have like 12% of inborn errors of metabolism which are covered by new therapies. However, 38% of these rare diseases are malignant disorders. And this is a little bit a problem in our field because the industry found out that it's very attractive to go to rare diseases, and to make a common disease rare is very simple with genetic tests, and then you can go to the market easier at lower costs and higher speed. And this is why we think there will be a rethinking how rare diseases are defined, so that the money that is should go to the rare and neglected disorders is more really to these rare ones. So as some of the goals have been achieved already ahead of time, there are the new goals now until 2027. And there's one particular problem that maybe all of you know that deal with patients with rare genetic disorders. It takes sometimes very long until the diagnosis. We have families with histories of 15 years until there's a diagnosis. Sometimes it takes like 3, 4, 5 years, and in all this time we are losing child health. We don't go for opportunities that we would have if we have the diagnosis earlier. And this is why there's one goal to diagnose a rare disorder at least within 1 year. Sounds long time, but still it's an optimistic goal. Now we want the IRDIRC says 1,000 new therapies should be developed. Okay. This is, 1,000 new therapies, this is a good goal. But if we think about that we have 8,000 disorders, there's still a huge need that we have to fill. And I would like now to focus a little bit on the topic that you have mentioned, the molecular basis of loss of function in genetic diseases. And just some background here, we know that we have 8,000 different genetic diseases, roundabout. This number will be fixed in maybe 5 to 10 years, and more than 20,000 disease-causing genes. When I was looking last time into the human gene mutation database, there have been around about 150,000 mutations known, and more than 60% of them are missense mutations or small insertions or deletions. And there's one common thing about these mutations. They all lead to the production of a protein. And normally genes encode proteins. Of course there are exceptions, but most of the genes encode for proteins. And if I have a genetic disorder here, there's still a protein produced. So if I think about the missense mutation or the genetic defect, and think about how the dysfunction works out, or what is the mechanism of dysfunction, we have to think about the loss of function of the protein. What is the problem in the protein then? And we ask the question whether or not there are common mechanisms to specific types of genetic variation. And we focused on missense mutations that encode the full-length protein, where I just have a replacement of one single amino acid in the full structure of the protein. And the good thing here, and we are talking about drug development, the targets are available. When I have an early stop mutation, for example, or a frame shift mutation, there is no protein anymore. So if I want to treat this problem, I maybe have to go to the genetic basis. But if the targets are still available, I can try to tackle them. This is just a short background, maybe a little bit of propediatrics. If proteins are produced at the ribosomes mostly, they have to fold into a native state, because the function of the protein does not come, I mean, in the end it comes from the primary structure, but it is exerted by a three-dimensional structure. So the structure defines the function, and protein has to undergo different folding intermediates until it reaches the native state. You can see it here in this funnel-like structure. And these folding intermediates have energy minima, and then they have to overcome them again to here be in the minimal best thermodynamic state. However, on the other side, proteins can get stuck here and in partially folded states, or they can aggregate. We all know the story about these amyloid disorders like Alzheimer and Parkinson and maybe prion disorders. And we have little helpers in the cells. These are the molecular chaperones, or known as heat shock proteins, for example, and they help to prevent the misfolding and to go to the native state. However, this does not always work out. And we have been working on this story at using one example, and this was phenylketonuria. And the reason why was a drug. We first had the drug, and then we had the mechanism. And I will show you how. So just shortly to the background for phenylketonuria, it's an inborn error of metabolism, and if the protein, the phenylalanine hydroxylase, does not work out, we have elevated blood phenylalanine in the patients. And this elevated blood phenylalanine is the problem because it somehow destroys the brain, which I will not focus on today. Mutations in this gene, or side chain replacements in the protein, can virtually affect all amino acids of the protein. And there's no hotspot. Doesn't matter where it's affected, it gets damaged. The physiological function is to convert phenylalanine to tyrosine, so one amino acid to the next one. And this is very interesting, it is regulated by its substrate. The substrate is an allosteric activator of the protein, meaning when there's a lot of substrate, the protein needs to be active, and then it's activated by the substrate itself. But there's also a cofactor, and this cofactor, tetrahydrobiopterin, in short BH4, is an inhibitor. And this makes sense because phenylalanine is an essential amino acid. So if we are not eating, we still need some phenylalanine in our blood. And if the protein would work at full capacity, maybe we would get a shortage of phenylalanine. So then the cofactor would inhibit the protein. And this is really a well-balanced and nice situation made by nature. Just to be precise, it's a homotetramer, this protein, 4 subunits of 52 kilodaltons, allosteric regulation I just explained, and the binding site, the second binding site for phenylalanine is in the regulatory domain and activates the protein. The current treatment, so phenylketonuria was the first inborn error of metabolism that could be treated, and the treatment came up shortly after the Second World War. And this is a low phenylalanine diet. And why did this come up shortly after the Second World War? Because in the Second World War, proteins were really needed, and people found methods how to separate amino acids to produce artificial proteins, so proteins for diet. And with this method, it was possible to have an amino acid mixture without phenylalanine because you could take it out over charcoal. And so having this low phenylalanine diet, you can tackle the problem. If elevated blood phenylalanine is the problem in the patient, the idea was very simple, to give lower amounts of phenylalanine to the patient, and then everything is good. This is true, but only partly true, because this diet is very restricting. This means that a patient suffering from a severe phenylketonuria can have per day, I mean, I'm exaggerating, but like 3 apples and 2 carrots. And if he would eat more, there's too much phenylalanine inside. So all protein intake comes from amino acid mixtures, and as you can imagine, this is not really healthy. Secondly, patients really suffer from the fact that they cannot eat like the others, and this is not a lifestyle diet. Patients, whenever they can stop their diet, they do. And we now know that even adults should not stop their diet because the brain is still under threatened and damaged by the high phenylalanine, and they're really looking for a new treatment. And then pharmacological doses of BH4 15 years ago came up as a new treatment. This was just an incidental finding. When BH4 was given to these patients, my wife, for example, and other researchers found out that phenylalanine in the blood is reduced, although the patients do not have a lack in the cofactor. So then the question is, if they don't lack the cofactor, but giving the cofactor has an effect, where does this come from? As I said, proteins exert their effects over their 3D structure, and of course, to be active, a protein has to move. If a protein is completely fixed, it is absolutely inactive normally. And we analyzed the structural changes that happen when the substrate and the cofactor bind. And if you look at the hydrodynamic radius of the protein by dynamic light scattering, you see an increase in size when the substrate binds. This is the activated or R state. If you give the BH4, and this was now the new finding, you see a decrease in size. So you have this T state conformation of the protein when only the cofactor binds. If first the substrate binds and then the cofactor, which happens in the normal conversion of phenylalanine to tyrosine, you are still in this activated state, which is needed for activity of the protein. However, if first BH4 binds and then phenylalanine, the protein stays in the T-state conformation. So it adopts a stabilized state, and BH4 prevents the protein from the conversion into the destabilized R state. We have analyzed many different mutations to look at structural changes, activity, and so on, by method like oligomerization with size exclusion chromatography, BN page, thermal shift assays. Maybe I will show a little bit here this thermal shift assay, where you see here the wild type in blue and then 2 mutations in yellow and red. And if you increase the temperature, the protein gets unfolded. This is what you do when you boil an egg. And if the protein is destabilized, the unfolding happens at lower temperatures. So this is indicative for destabilization, and we can have this as an endpoint in our drug discovery campaigns later. What we found out is that mutations lead to altered allosteric regulation. So we know that allosteric regulation is very important for protein function, and every mutation that I have analyzed so far in this gene leads to allosteric alteration of the allosteric regulation. We have conformational destabilization, aggregation, reduced stability against heat stress, as I said. So in the end, a loss of thermodynamic and kinetic stability or which can be translated as a loss of structural dynamics of the protein and its stability. We did these studies on the PAH protein in Phenylketonuria. However, many researchers worldwide were working on other proteins and found the same problems. Very renowned examples are morbus Gaucher and morbus Fabry with protein misfolding, and on the other hand side, cystic fibrosis, which is maybe not a neurological but more lung and gut disease. And for some of these disorders, drugs have already been developed. So for morbus Fabry, for cystic fibrosis, for phenylketonuria, we have pharmacological chaperones in hands. For others of these targets, projects are running. And in our lab, we have been focusing on in total 6 different disorders where we could show that protein misfolding is the molecular phenotype in these inborn errors of metabolism. Besides phenylketonuria, we focus a lot on MCAT deficiency, is the medium chain acyl-CoA dehydrogenase deficiency. It's a degradation disorder of fatty acids in mitochondria, which has the same incidence as phenylketonuria, 1 in 10,000. So we have 50,000 patients only in the EU, for example. To sum it up, the molecular mechanism how missense mutations lead to dysfunction and loss of function can be inefficient protein folding and accelerated protein degradation. However, you always have to keep in mind that there are certainly some mutations that affect the catalytic center of the protein. So this protein can be nicely stable and not degraded, but just completely inactive because mutation hits the active center. However, the active center is rather small compared to the full size of the protein, so these mutations are rather rare, however they are there. If we now know what happens when missense mutations affect proteins, how can we rescue protein misfolding? What could be a mechanism? We have analyzed and focused a little bit on mutations at the interface of the subunits in the protein, here depicted in green. And we looked at the conformational changes that take place when substrate and cofactor phenylalanine in blue and cofactor in orange bind to the protein. And we analyzed the fluorescence of tryptophan 120, and as you can see, whenever BH4 binds to the wild type or to the mutations, you see a decrease in tryptophan fluorescence. So you see that there are conformational changes that take place when this cofactor binds to the protein. However, if you use an artificial mutation here where you cut off the regulatory and the oligomeric part of the protein, just stay with the catalytic part of the protein, the conformational changes are reduced. So the protein needs to be a tetramer, the protein needs to be able to do the dynamic changes so that BH4 can exert its conformational rearrangements. The same goes for hydrophobicity, which I will not focus on, and also with dynamic light scattering. Just one example here is the mutation D415N, and you can see that this mutation is compared to the wild type already almost in the R state. And when phenylalanine binds, this R state is really increasing, so the protein gets a very big size and is very nicely accessible for degradation enemies, I would say, in the cell. And when BH4 binds here, it's really put back to the T state. So what is the effect of BH4 on a molecular basis? We analyzed thermodynamics, so we analyzed the let's say, a thermodynamic basis of the unfolding process, and we could see that there's no shift in free energy, but there's a shift in the activation energy, here depicted as EA. And if you think of your basic studies, every chemical process needs an activation energy to go from one stage to the next stage, and if we analyze from the folded to the unfolded stage, BH4 increases the energy that is needed. So this means that there is a thermodynamic protection when BH4 binds, so the protein rather stays in the folded state. If this is true, very simple test, we can try to heat up the proteins and see whether BH4 helps to reduce the temperature, helps to increase, the temperature that is needed for the unfolding process so that the protein is more stable. And we see for many different mutations on the x-axis you see the site of amino acid exchange, and on the y-axis the temperature. You see that BH4, the black circles, increase the temperature, so there's more stability when BH4 binds. Then, meanwhile, this drug was already on the market, BH4 as a treatment for PKU. The drug was on the market even without knowing how it works, because the clinical studies have been so impressive that FDA and EMA said approval is okay, even without any animal model, nothing. Still, we wanted to find out how it works out, and we used in green depicted the BH4 responsive mutations, so patients carrying these mutations responded to the drug, and we can say that these mutations, if expressed in purified proteins, really nicely respond to the addition of BH4 and lead to a steep or huge increase in temperature stabilization. Of course, we showed it in the mouse model, what happens if we give BH4 to the PKU mouse model, which is called ENU1, and you see elevated blood phenylalanine if no drug is available, but if the drug is given to the animal, the blood phenylalanine goes to the normal level. Then we developed an endpoint, because we are not so much interested if we talk about rescuing the protein, and we only have the phenylalanine as the biomarker, there's a huge gap in between. So we decided to try to have a marker for the protein itself, and we developed a breath test, 13C phenylalanine oxidation test. So we have this stable isotope, 13C, in the phenylalanine, and if the animal metabolizes this phenylalanine, it has to be due to the activity of the PH protein. And here we see the cumulative recovery of CO2 in the exhaled air. For the wild type, it's high, and for the animal model, it's low. And this is another animal model which is not responsive to the drug, in black. And you see up on the addition of BH4, you have the correction of the metabolism to the wild type level. But some of you who are really looking nicely at this graph will see that there's a difference even for the wild type. And this is the inhibition I was talking about of this BH4 molecule. But for the wild type it doesn't matter, because the activity is far high enough. So if you inhibit from 100% to 80%, no problem. But if you increase from 5% to maybe 20% of activity, you have a normalization of the metabolism. And this is why this trade-off of inhibition and stabilization works out for the patients. Yes, that's the gap. Pharmacological chaperones, just to sum it up as a take-home message, these are small molecules that stabilize correct folding and lead to rescue of protein function. So targets must have a mutation which renders the protein unstable but not completely inactive, like a mutation in the active site of the protein. You cannot stabilize by pharmacological chaperones because it's just inactive. PC pharmacological chaperones bind specifically and with high affinity, so that they can be used at low concentrations, and this is important for future drug development processes and initiatives. Inhibitors, as I showed, are good candidates for pharmacological chaperones, and specific inhibitors work better than non-specific stabilizers, I would say. So many people are doing proof of concept experiments with high glucose, for example, but you cannot put the patient in at 20% of glucose, which works nicely for proteins, but maybe not for patients. So pharmacological chaperones are effective if a small amount of the protein can make a big difference. Now, having understood this in our lab and with many other labs in the world, we said that now we have to translate it, because it's good to have one drug, but if we understand that this is true for also other disorders, we maybe should go and try to develop drugs ourselves. However, there was still one problem. BH4 works nicely for PKU patients, but only for 30% of them. So 70% of the patients are still untreated. I mean, they're treated by a low phenylalanine diet, but they don't have a drug alternative. So, how to approach it? One approach would be hypothesis-driven medicinal chemistry. You take, for example, here the BH4, and then you introduce changes in this molecule and would improve it, and maybe it binds to also the mutations that do not respond. We did not do this because we don't know how to do. And we did not find partners that would love to go into this direction without getting a lot of money from us, which we don't have. So what we could do is hypothesis-free high-throughput screening, just to look whether there are new compounds that would bind to the protein and stabilize it. And this is what we did. We established first 3 platforms. One platform for functional testing, because this is the endpoint. You want to increase the function in the patient, so you need to have excellent assays for this, not only in the test tube or 96 well plate, but also later in the patient. And we have it with this breath test, which we can do with the animals and also with the babies, because to exhale is very simple, and every organism does it. We have established a platform for high-throughput screening to find the new drugs, and we have established a platform for hit validation, so orthogonal assays where we can then prove that it's really this molecular mechanism and find out the binding affinities and so on. And this is already the last, this is the last part of the talk. I would like to show you just in brief what our approach is. First, we do a virtual screen. We have the 3D structure of the protein. So we screen 8.8 million compounds, whether or not they could maybe bind to the protein. And then we select around 300 hits. We have certainly more hits, but this number 300 has to do with the capacity of our laboratory and our machines. And with 300 hits, you can cover a nice chemical space. Then we perform with the wild type and variant proteins the screen. In industry, often only the wild type proteins are used for the screen, and the variant, so the mutation, the disease model, comes later. We think this is wrong, or it's not the best way for this approach here, because the variant proteins need the treatment and they need to be stabilized. So this is why we already have them in the screen. We do 3 methods in parallel. So thermal shift assay, as I showed you, then a functional assay on activity and protein amount, and a binding assay. And this is our result sheets. Just as an example, we have the wild type here and 3 different mutations at 3 different domains in the protein, and you see that more than 30% of the compounds that we selected are hits. And this is okay because we selected them for binding. And if they are binders, they would have some effect on this thermal shift assay, because every binding leads to an increase in the temperature or an increase in the stability. However, we defined we need a strong effect, so more than 2 degrees and not 0 point something. And doing this and doing cluster analyses to find the best hits, we still could select 50 different hits. Going to the next method, the Surreal Screen, you already see that there are the number of hits is lower, because this is already our functional endpoint. But when we combined the activity and protein amount that we were looking for in the cellular screen, we had 67 hits. And then we could identify 6 groups with, by effect size and type, with 3 most promising groups and 4 to 5 lead structures. Or in a short summary, we performed 3 different screens, found for all 3 methods different hits, but there was an overlap, and we were very happy that in the end, in the middle here, there was not a 0, but a number. And this was a screen we did for the PKU, and at the right side, this was a screen we did already for the next disorder, for MCAT deficiency, and the third one is currently running for glutaric aciduria type 1. And we think that this approach to use different methods in parallel and then go to the area where you have the best overlap is very nice to come quickly to lead constructs, lead compounds, sorry. And now maybe Dana could come into the play and help us with translating this into the real drug development process, because from here to clinics, there's still some steps needed. You need to analyze the toxicity, of course. You need to better understand the binding behavior, pharmacokinetics, and so on. We think that from here to the clinics, an amount of maybe 800,000 to 1 million euro would be needed. And we were trying to find possibilities for funding from academia. This did not work out, so we funded a company and did a little roadshow, went to different companies and presented what we have, and the response was overwhelming. So we will have no problems to go these next steps. So the money is there out, and maybe sometimes we have to just move from academia out where the money is, where the biotechs are, and work together with them. And I'm a children's doctor, so for me it's not natural to walk over to the biotech companies and say, here, I have something, don't you want to bring it to the market? But obviously they are willing to do so, and if we can convince them that we have good targets, that we have good ideas, and that we have good assays to go later to the clinic, which is very, very important for them, for the clinical trials, maybe then we will be able to do academic drug development, or academically initiated drug development at least. At the end, I would love to present people who contributed on this long journey. Mostly for the screen that I showed, Anna Waldhuber did it for PKU and for MCAT deficiency, and Madalena Barroso now takes over because Anna left our lab and went back. We moved just recently from Munich to Hamburg. This is more than 800 kilometers and completely different cultures in Germany. And now Anna moved back to Munich, because in Munich you have Oktoberfest. This you don't have in Hamburg. And it just ended yesterday. Madalena came from Portugal, so she didn't mind whether she is in Munich or Hamburg because it's too cold anyhow. And I would love to thank all the other people that helped in this journey, and certainly Anja, who is the founder of this all this research, plus my wife, and thank you for your attention.
Thank you so much for such an exciting
Do you need a microphone so that people... I don't know whether it's switched on, but...
Now it's working.
Perfect.
Yeah. As we discussed in the past, I really like this concept of misfolding connected to inborn error of metabolism disorder. And I think that in the case of PKU, it's very compelling, the evidence that you provide. This model was tested in other diseases, let's say giving some chemical chaperones in other inborn error of metabolism disorders. that is not related to the mechanism whatsoever, can you see any beneficial activity?
Yes, this problem, protein misfolding in inborn errors, not only inborn errors of metabolism, has been shown now in 60 different disorders as far as I know. I'm trying to keeping track on PubMed. However, maybe I missed some and it's already 80. There is not only evidence by, for example, chemical chaperones, but there's already new drugs on the market for Morbus Fabry now. I think it's a very promising target to go for. Many of these disorders are neurological disorders, for example, Allosteric aciduria type 1. I think we and others showed how you can go the way. It's not very complicated. It took us a lot of money to set it up. Yeah, the full screen I showed here was 2 million euro. I would say the next time we do it, it would be like 500,000 euro because the money was needed to set it up. And if we do it again and again, we can reduce the costs and have more disorders in the screen. And also, I would love to encourage you because if we can do it, you can do it anyhow.
More questions? Yes?
Yes, very important question. So the first one was how can we define out of 10 million or 8.8 million compounds, what is the good ones for us? Yeah, we define we know the binding site that we are interested in. Sometimes we even define some new binding sites. And then we just, by a virtual screen, look for compounds that bind. That's all. Yeah, it's only binders. Then comes the assays that we do. And as in our assays, we have functional endpoints that are related to misfolding and activity. I think then we can rapidly take out those that have the activity that we need. If we have completely new compounds, of course it's very complicated to make sure that there are no side effects. You cannot do that. In this library of 8.8 million compounds, there's a lot inside. However, the 300 that we select are pre-selected for not being arsenic compounds, for example, or something that we know that certainly would kill the person. So there's this Lipinski rule of 5, for example. If you follow this, you know that there shouldn't be too much toxicity, there shouldn't be too much lipophility, and so on. However, of course, you have to go for ADME-tox screens to find out whether there are side effects or not. And the first drug that was developed, and we were part of it, was this BH4, natural compound of the liver. Now we have it in the pill, just at higher doses. What was the risk? I would say not 0, but 0 point something, that we would have problems. We don't have problems. Now we have lead structures that we find, and my biggest fear is that one day you will read in the newspapers that a crazy doctor from Hamburg was poisoning kids. So yeah, certainly. I mean, I studied medicine to help, and now I find out that there are many disorders where I don't have anything in hands. So I think we have to be, we have to encourage ourselves to go that risk. There are many pharmaceutical companies that know how to reduce this risk, so we don't have to invent it. But still, the risk is there. I wouldn't be honest if I wouldn't say it. Tja, this we will find out later.
Thank you. More questions? A small question I would like just to ask if your screening platform has a capability to optimize for drugs that should penetrate blood-brain barrier and will be useful for neurological disorders?
So, just one thing, PKU is a neurological disorder. However, the target is in the liver.
Yeah.
So sometimes the targets are outside the brain,
Yes.
which is funny enough, but it takes place. Yes, we do have some parameters for this. However, we could always benefit from ideas how to screen for that. And if you have good models, we are looking forward to hear about it. Because, for example, glutaric aciduria type 1, this is a disorder that affects an enzyme which is expressed highly in the liver, but still also in the brain. And we think that we need to address both targets, the brain target and the liver target. And it's expressed in the kidney, to be precise. Yeah.
So I would like again to thank Søren for just a great talk, and I think we are talking about collaboration between BLAVATNIK, Sagol and Hamburg, so it will be really great. Thank you.